Let's begin by introducing the polymerase chain reaction, or PCR. PCR is a revolutionary technique in molecular biology that allows scientists to make millions of copies of a specific DNA sequence in just a few hours. This process is performed entirely in a test tube, making it accessible and efficient for laboratories worldwide. PCR has transformed research, diagnostics, and even forensic science, enabling the detection and analysis of genetic material with unprecedented speed and accuracy. In this video, we'll explore how PCR works, its history, and its profound impact on science and medicine. The story of PCR begins with Kary Mullis, a visionary scientist who conceived the idea in the early 1980s. He introduced PCR to the scientific community in October 1985, presenting it as a simple yet powerful method for producing virtually unlimited copies of a specific DNA sequence. Before PCR, amplifying DNA was a laborious and time-consuming process. Mullis's innovation changed everything, opening new doors for genetic research, medical diagnostics, and beyond. His contribution earned him the Nobel Prize in Chemistry, highlighting the significance of this groundbreaking technique. At the heart of PCR lies the principle of DNA hybridization. This fundamental concept in molecular biology refers to the complementary pairing of DNA bases adenine with thymine and cytosine with guanine. PCR exploits this chemistry to ensure that primers bind precisely to their target sequences, setting the stage for accurate DNA replication. Understanding DNA hybridization is crucial for grasping how PCR achieves such specificity and efficiency in amplifying genetic material. PCR functions as a test tube system for DNA replication. Its main purpose is to selectively amplify and enrich a specific DNA sequence, known as the target, by several million-fold in just a few hours. This remarkable amplification is achieved through a series of carefully orchestrated steps, each designed to maximize the yield and fidelity of the copied DNA. The process is both powerful and precise, making it indispensable in modern biology. Central to PCR is the enzyme DNA polymerase. This indispensable enzyme is responsible for synthesizing new DNA strands by adding nucleotides to a growing chain. In PCR, DNA polymerase targets a specific fraction of the genome, guided by short DNA sequences called primers. The enzyme's ability to withstand high temperatures is crucial, as PCR involves repeated cycles of heating and cooling. Without DNA polymerase, the rapid and efficient amplification of DNA would not be possible. Primers are short DNA sequences that serve as starting points for DNA synthesis in PCR. Two different primers are used to bracket the target region. One binds to one strand at the beginning of the target, while the other binds to the opposite strand at the end. This arrangement ensures that only the desired DNA segment is amplified. The specificity of primer binding is what gives PCR its remarkable accuracy and reliability. The PCR process consists of repeated cycles, each with three main steps: denaturation, annealing, and extension. During denaturation, the double-stranded DNA is heated to separate it into single strands. In the annealing step, primers bind to their complementary sequences on the DNA. Finally, during extension, DNA polymerase synthesizes new DNA strands, extending from the primers. These cycles are repeated multiple times, leading to exponential amplification of the target DNA. The first step in each PCR cycle is denaturation. Here, the reaction mixture is heated to around 94–98 degrees Celsius, causing the hydrogen bonds between DNA strands to break. This results in the separation of the double-stranded DNA into two single strands, making them accessible for primer binding in the next step. Denaturation is essential for resetting the DNA template, ensuring that each cycle begins with single-stranded DNA ready for amplification. Following denaturation, the reaction mixture is cooled to a lower temperature, typically between 50–65 degrees Celsius. This allows the primers to bind, or anneal, to their complementary sequences on the single-stranded DNA. The precise temperature is critical. Too high, and the primers won't bind. Too low, and non-specific binding may occur. Annealing sets the stage for the DNA polymerase to begin synthesizing new DNA strands in the next step. In the extension step, the temperature is raised to around 72 degrees Celsius, which is optimal for DNA polymerase activity. The enzyme attaches to the primers and begins adding nucleotides, synthesizing new DNA strands complementary to the template. This step is where the actual copying of DNA occurs, and it's repeated in every cycle to exponentially increase the amount of target DNA. One of the most remarkable features of PCR is its exponential amplification. With each cycle, the number of DNA copies doubles, leading to millions of copies after just 20–40 cycles. This efficiency is what makes PCR so powerful for detecting even tiny amounts of genetic material, whether in research, diagnostics, or forensic investigations. PCR is a quick and reliable method for detecting mutations associated with genetic diseases. It can identify a wide range of genetic changes, from insertions and deletions to single-point mutations. This capability makes PCR invaluable in clinical diagnostics, allowing for early detection and management of genetic disorders. Beyond research, PCR has become a cornerstone in clinical laboratories. It's used to detect tiny amounts of pathogens, such as the human immunodeficiency virus, HIV, and to diagnose numerous other genetic anomalies. The sensitivity and specificity of PCR make it a trusted tool for doctors and scientists working to improve human health. PCR's ability to amplify tiny amounts of DNA has revolutionized forensic science. Even trace amounts of genetic material from a crime scene can be analyzed, helping to identify suspects or exonerate the innocent. The reliability and speed of PCR have made it a standard technique in forensic investigations worldwide. In summary, the polymerase chain reaction is one of the most significant innovations in modern science. Its ability to rapidly and accurately amplify DNA has transformed research, diagnostics, and forensics. PCR continues to evolve, with new variations and improvements expanding its applications. As we look to the future, PCR remains a cornerstone of molecular biology, driving discoveries and improving lives around the world.